All right, so now we're going to look more in depth at an action potential. This is really, really important. It's really good information. All right, so what does it mean to be polar? The cell membrane we're going to be talking about is polar. So polar means basically, think of the North and South Pole. They're kind of opposites, right? Or if you think about a magnet that has a positive and a negative end, that's polar. And so the cell membrane is electrically charged, or what we call polarized, because on the inside of the membrane it has a slight negative charge. And you can see here intracellular means inside, and this is that slight negative charge, and this is at what we call at rest, when there's not an action potential being fired. And the extracellular environment, or outside of the cell, tends to have a positive charge when it is at rest, kind of. It's not typically, truly at rest, but that's just the best way to describe it, okay? And so the inside is negatively charged with respect to the outside. And this is the result of the ions that are inside and outside not being distributed in an equal portion. All right, and so on the outside of the membrane, you're going to have a higher portion of sodium ions, and on the inside you have a higher portion of potassium ions. You also have chloride ions and some other things in there as well. Okay, and so it's important to understand what we call, this is the resting potential when an action potential is not occurring, because we're going to look at the changes that occur during an action potential, and those changes are what allow us this basically domino effect to occur that sends that signal on down the membrane. All right, so let's look a little bit more closely at the distribution of ions here. The ions, the distribution, I should say, is maintained via active transport. And so these ion channels here, help move sodium and potassium across. And we have different channels. We have gated ion channels, we have leak channels. So we have things that move by active transport or things that move by simple diffusion. And so active transport is allowing the sodium and potassium ions in and out. And this often occurs in response to that action potential. But we also have some slow leak channels that allow one through at a time. But more importantly, we have the sodium-potassium pump. And what it does is it shuttles 3 sodium ions out, okay, and 2 potassium ions in. All right, and that kind of helps restore and keep the balance, especially after an action potential occurs. So active transport helps maintain a greater concentration of sodium ions on the outside. That is important, that's a hint. And potassium ions are greater on the inside. You need to remember that. So the inside of the membrane here has an excess negative charge. It is more negative. And the outside has a more positive charge. Now we say more positive or more negative because if you have a number like negative 70 and a number like negative 20, negative 70 is more negative than negative 20. Okay, and so as it's changing, it might still be negative, but it's becoming more positive, closer to 0. All right, which can be kind of hard to wrap your mind around if you don't just stop and think about what a negative number is, and think about the number line here, 0, and I'm 20 from 0, or I'm 70. You can't see my hands here. I don't know which way to go to get you there. 70 from 0. All right. And so this is what we call the resting potential membrane. That is very important for you to remember. Resting potential membrane, and this is when the action potential is not occurring, okay? All right, so as ions move from one side to the other, so if more potassium ions move in, the charge is going to increase, right? The positive on the inside of the membrane is going to become more positive. And it only takes a few ions to move in for a change in the membrane to actually show. And if potassium ions move out, you're also going to see the inside become a little more negative. All right, so it depends on which way they're moving. Even though we only have positive moving, we can still make something more negative by taking positive away. All right, so only a small amount of ions are needed. You can think of that as if you have 1 grain of sand in your eye, it is a pain. But if you have 1 grain of sand on the beach, you can't even tell, right? So 1 grain of sand in the eye is big, causes a big change. All right, so we already talked a little bit about resting potential and kind of touched a little bit on action potential. So let's look at those, and we're going to add in a graded potential. Graded potential you need to understand, but resting in action is really the most important, okay? And so resting potential, that's when the membrane of the cell, we say undisturbed, even though technically those things are still happening, but we have that resting potential membrane. It is more negative on the inside, more positive on the outside, has a higher concentration of potassium on the inside, higher concentration of sodium on the outside. All right, so when a change to that resting potential membrane occurs, that's when the neuron starts getting to work and gets busy. Okay, and so dendrites, remember, these are receivers. These receive information over here. Okay, and so some stimulus is going to produce a change in the dendrites, in the localized here resting membrane potential. The change decreases the further away it gets from the potential, and here we call this just a graded potential. Okay, an action potential is when the graded potential reaches a threshold amount. It has to reach a certain number to have enough energy, so to speak, to make that fire. Okay, the change, once the action potential develops in one location, once it starts, once you pull that trigger back and squeeze it, when you squeeze that trigger, you can't stop it, it's going to fire, right? So you squeeze that trigger and it goes. Okay, so it's going to spread along the surface here and keep going. If you have a line of dominoes and you push the domino at the very front, and if they are spaced perfectly apart, are you going to be able to stop them from falling? No, they're all going to fall. Okay, that's basically how an action potential occurs. It's like that triggering of the dominoes falling. All right, so let's look a little more at resting potential. Remember, this is when an action potential, when the action is not happening, so there's no action going on. Truthfully, there's a little bit of action, but not much. Not an action potential. There's things flowing in and out, but there's no message being sent. So when an ion goes to the outside, which ion is going to have a higher concentration outside the cell? I told you that a minute ago. If you said sodium, you're right. Which one has a higher concentration during the resting potential inside the cell? That is potassium, right? And so which side has the greater, more positive charge, inside or outside? If you said outside, that's extracellular, you're correct. And so the intracellular, or inside the cell, is more negative. Okay, so I hope you're kind of thinking that along with me as I'm saying it. We call this a potential difference, okay, because the outside has a higher charge relative to the inside. One way to kind of remember where the ions are is to think of a neuron like a salty banana. Bananas are good for potassium, right? So there's lots of potassium inside, and we have on the outside, we have salt, okay? There are some chlorine ions in here that are negative, but we don't worry about those too much right now, all right? So salty banana, salty on the outside, lots of good potassium on the inside, all right? So, like I said, we do have some slow leak channels that allow one particular ion to go through at a time, and then we also have that active transport going on, all right? And they're going to go, of course, according to their concentration gradient. A potential is a separation of charge. That means the charge is not equal across the membrane. That movement through the potassium leak channels, which allow potassium to move out down its concentration from high to low causes a separation of charge and the appearance of an electrical gradient, because you have more negative on one side and more positive on the other. Okay, so leak channels only let ions go through one at a time very slowly. We also have sodium leak channels as well. And again, of course, we have that sodium-potassium pump that moves 3 sodium out for every 2 potassium ions. And that helps to keep up with the leak channels to maintain that balance. All right, so that's resting membrane potential when no action potential is being fired. All right, so we're on resting potential here. We do have channel proteins in the membrane. The enzymes also that we need, like the sodium-potassium pump, is also called sodium-potassium ATPase, is an enzyme, and this is found in the plasma membrane of all animal cells. It has several functions in cell physiology. All right, we have sodium and potassium pumps, and we call these gated channels. So let's look at the difference of these real quick. What color? I think you can see the best with maybe red. All right, and so this leak channel is kind of always open. All right, it allows basically one ion to go through at a time, and it is specific to the ion. So it's only going to allow potassium, and I drew the arrow the wrong way. Sorry, it goes that way. Potassium to go out. All right, and then here's a chlorine or chloride leak channel, allows chlorine to go out. And then you have these gated channels, and see how this is closed. And so something can trigger that to open and allow sodium to go in, or it can be closed. Okay, so leak channels are always open, but gated channels need something to open the gate, and that is important. All right, they need something to open the gate. And then we talked about the sodium-potassium pump moving in and out, and it is always active. All right, so this is not my favorite diagram, but if you really look at it and try to understand it, it is helpful. Okay, when the membrane of a cell is undisturbed, what is that called? Resting membrane potential. It reads about -70 millivolts. Okay, -70 millivolts. All right. So that sodium-potassium pumps sends those ions back and forth to restore the concentration. It uses ATP to move the ions against their concentration gradient. All right, so they're going against their concentration gradient. So the membrane is polarized. The inside of the cell is negative relative to the outside of the cell, and again, that's due to the distribution of ions. All right, and then it's restored by the sodium-potassium pumps. And so here in A, this is showing you a neuron before the membrane potential is established. All right, potassium ions are leaving the cell. You can see that here. Oh, potassium ions leaving the cell, sodium ions going in. All right, and you don't see any difference in charge. Okay. Now over here at B, when more sodiums go out, that loss of positive charge in the cell is going to cause that distribution change, and so it's going to be more negative on the inside and more positive on the outside, causing what we call a potential difference or an electrochemical gradient. All right, so slightly positive and -70 is the typical neuron. All right, for resting membrane potential. So let's look at these gated channels. What causes them to open and close? You can have changes in the environment in the cell that can cause those gated ion channels to open. If you have more positive ions flowing through the membrane, it's going to change the distribution of charges. Okay, and so it's going to cause that membrane potential. If a membrane becomes more negative, we call it hyperpolarized because that difference is larger. If the membrane potential becomes less negative, we call that depolarization, meaning we're bringing those charges together and they're no longer going to be polar. Okay, so the difference has decreased. You must reach threshold potential again in order for an action potential to be triggered. Okay, threshold potential, that is really important. Threshold potential triggers those voltage gated channels to open. So when we say voltage, we mean actually electrical voltage. Okay, and so those graded potentials help stimulate or reach a threshold potential. Threshold potential is a high enough charge that it triggers those gates to open. Okay? If it is not at the right level, not at the right threshold, the gates will not open and an action potential will not be fired. But once you fire that, once you push the top of those dominoes over, it's going to be triggered. Okay? And so that electric current is what's going to cause those gates to open. Now, there are other things that can open gates as well, okay? And this is important because we're talking about an action potential, we're talking about electrically gated, or voltage gated, but there are other ways that this can happen. Basically, gated means there's something that's going to cause it to open. And so we can have voltage gated, which we just talked about. You can also have chemically gated or mechanically gated. And so there can be other stimuli to cause these to open, like changes in temperature, changes in actual pressure and feeling of something, and changes in amounts of light, okay? And this is going to cause the channel to actually change shape. All right, and then that's going to open up enough to allow ions to pass through. So these are our voltage changes, and we've kind of talked about these. Resting potential, there's no change, but we have our graded potential and our action potential. A graded potential can have different strengths, and we can add those together or sum them. Okay, and remember that's on the end down by the dendrites in the cell body. These can only travel a short distance. They can lose strength as they travel, and we use them in short distance communication. If they are summed together and strong enough, they can come together to trigger an action potential. The action potential will always be all or none. All or none, okay? So you could think of the graded potential, I guess, as putting the dominoes in their places. I mean, that's kind of not the best analogy, but I don't know, maybe you're getting ready, let's look at it this way, you're stretching and warming up before the race, okay? And then the action potential is when you're running the race. I think that's a little bit better analogy. All right, so the action potential, though, is all or none. You cannot add these together. It either fires or it doesn't fire. It doesn't lose strength. It has the same strength all the way across. Whatever amount of pressure you push that domino over with, you are going to have that same force go all the way through, right? And so you need that threshold stimulus. You can and it would be hard to do this, but you could just very, very lightly touch the domino and have it not topple over, but you have to push with a certain amount of force for that to overcome static friction, which means the force that you need to make it move, okay? And so that is really important, that you have to have that threshold stimulus to get it started, and once it triggers, it's going to go with the same all the way, and it can go a very long distance. So this would be a really good diagram for you to draw out and label and be sure that you know this, okay? Often something that I use in, what's the word I'm looking for? Essay questions. Sorry about that. So the resting membrane, we already said, has a potential charge of -70, okay? And so here we have our threshold line at -55. So we need the voltage to go from 70 to -55. So it needs to change by +15 millivolts, okay? It will not begin until it reaches that mark, okay? When the stimulus occurs that reaches that threshold value, the voltage needed to cause the action potential to fire, okay, then it is going to be that all or none. It's going to finish. So once it gets to here, reaches this threshold amount, then this part's going to occur, okay? If it is below, if it's here, nothing's going to happen, okay? It has to be this much of a change in voltage, all right? It's got to reach that threshold stimulus, all right? And so that's going to come in. Sodium ion is going to cause the sodium ion gates, and I'm getting a little ahead, but that's okay. Repetition, repetition, repetition. The sodium ion gates are going to open. Sodium ions are going to rush in. And then as the sodium ion gates here close back, potassium gates open and potassium rushes out. Okay? And so that's what these numbers are telling us. All right? So we will have a stimulus that causes a threshold. It reaches the threshold and will cause the action potential to fire. Sodium ion channels will open and rush in, and we call that depolarization. Okay, it's becoming less polar. The charge is getting closer to the same inside and out because you're flooding more positive ions to the inside. All right. And then as those dominoes are toppling, the sodium ion channels close, potassium ion channels will begin to open. And so when potassium ion channels begin to open, potassium runs out. So that's decreasing the positive charge and causing that potential difference to reoccur, and we call that repolarization. Now, potassium ion channels are not completely in sync with sodium, so it takes a little bit of time for them all to close, and then the sodium potassium pump has to kind of catch up and restore that balance, and that is where we get what we call overshoot or hyperpolarization. So it takes a little while. Can I get my color back? It takes a little while to get back to normal. All right, and so that charge will go the opposite way for a little while, but then it will go on back to normal, mostly due to, like I said, potassium ion channels take a little bit longer to close, but mostly due to that sodium potassium pump restoring that balance back to resting state, bringing us back to normal. All right, so resting membrane potential here we have at -70. You've got more potassium on the inside, more sodium on the outside. So you can see we're at -70 here, we're at resting potential. So an action potential is fired. Actually I should start with, there's a threshold stimulus that occurs and causes the action potential to fire. And as this line is moving up, these start to flood in, all right? And as those start to flood in, those dominoes start to topple, okay? You can see these arrows are going in, and then the potassium starts to flow out, okay? And so there's where we're getting the repolarization, or the portion going down this way, and then you have the overshoot or hyperpolarization. All right, so what do we have here? Well, we have a presynaptic and a postsynaptic neuron, all right? So here is the axon terminal of the presynaptic neuron. As you can see, there are neurotransmitters being released into the synaptic cleft, all right, and then they're being picked up, and so they're causing a graded potential in the cell body, all right. So those ion channels are triggered by some neurotransmitter from the previous neuron. And so the cell has become more positive. So you can see it's gone from -70 to -62, all right, -62. So the cell is now somewhat depolarized, but an action potential has not happened, okay, because it has to get to that threshold amount, which is -55. Oh, -55, okay. And so again, remember the axon hillock is where the trigger zone is, where those changes occur. When enough sodium ions enter, okay, the charge is going to continue to change enough until it finally gets to -55 millivolts. And it's going to reach that threshold amount at -55. All right, and so as soon as it reaches that threshold amount, and I want you to look, I'm going to erase a little bit more. Look at this portion here. There's nothing going on, all right? No sodium going in. But once you reach that -55, sodium ion channels begin to open, and it's going to go all the way down the axon, okay? They're going to allow the sodium to flow in, which is then going to trigger the potassium to flow out. All right, so let's summarize. The first part of the axon is called the trigger zone. That's the axon hillock. Because it contains a whole lot of those voltage gated sodium ion channels. All right. Changes in voltage cause those voltage gated channels to open and allow sodium to flood in. Okay. So sodium is going to flood in. All right, and so the sodium channels then begin to close, and as they are closing, it triggers the potassium ion channels to open, and so potassium flows out. And so when the sodium channels first move in, you're changing that positive charge, that negative charge to be more positive, and so we call that depolarizing. We are separating, closing in that separation in charge. We are bringing it closer together. Then when the potassium ion channels open and potassium leaves out, potassium is positive, it leaves, so you are making it more negative, and that separation in charge begins to grow. Okay, and then so we call that repolarization. So depolarization brings the charges closer together. Repolarization brings them back further apart to make them polar again. All right, and so if this is resting potential, remember, as potassium moves out, we are going to hyperpolarize. Okay, a little bit. We're going to hyperpolarize because it takes a while for those potassium channels to close and the sodium potassium pump to bring it back to a normal resting potential state. Okay, so resting potential, depolarization, repolarization, hyperpolarization. All right, and that's active transport that reestablishes the resting potential. So this is showing you how those dominoes are triggered, okay? So that nerve impulse is those action potentials are being passed on from one domino to the next, all right? So if you'll look at this here as it's going down this direction, when these sodium ions go in, the potassium goes out. More sodium goes in, more potassium goes out. And so it's showing you how that impulse is being conducted all the way down. The propagation, which means to spread and promote. So the action potential is spread along the axon to the next section of the axon until it goes to the end and eventually to the next neuron. It's kind of like during the wave. When you raise your arms up, the neighbor next to you puts them up. As theirs are going up, yours are going down. It's very similar to that. If we were in class, I would actually have you stand up and do that, okay? But there has to be a stimulus. Somebody has to say what happens. If you've ever been to one of those games, there's somebody standing out in the field showing you what to do. Okay, so that's the trigger, that's the stimulus. So you got to reach that threshold amount for that action potential to start. So when it comes to neurons, we have the all or none law, and I've said this several times really already. An action potential is all or none response. It will either occur or not occur. There's no in between. It will respond completely. It doesn't depend on how often you get the stimulus, and you can't add them together necessarily. It's got to reach that threshold amount, all right? A nerve impulse is going to be conducted whenever that stimulus of threshold intensity is applied to an axon. All right, and then it is carried along the axon with the same strength. So the speed that those dominoes are toppling, it's stays the same all the way down. A greater intensity of stimulation will only trigger more impulses per second, not a stronger impulse. All right, so we touched on this a little bit in the very beginning. We're talking about the refractory period, and there's actually two. There's the absolute refractory period and a relative refractory period. And so I was talking about how when you have a Charlie horse, it hurts, right? And so you don't want the stimulus to be continually coming over and over and over again, stronger, 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 bigger, 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 right? That would not be very good. So for a short time during the firing of that impulse, the axon is unresponsive to a normal threshold stimulus. Like I said before, you have to recock the gun, okay? And so until that gun is recocked, you can't send another stimulus, all right? Except this is, and actually, I don't know how familiar people are with guns, but there's kind of like a little cock and then another little cock. I really don't know what the difference is between those two, but it's broken into two, and we have that here, okay? So the axon is unresponsive for a little bit of time. We have the absolute refractory period. Once the action potential has started, no matter what, you can't trigger another one, okay? That is absolute not going to happen, all right? The sodium ion gates are not reset. So that's why I said it's like cocking that gun. The sodium ion gates are not reset. The relative refractory period, a stronger depolarization would be needed. Okay, so this is if you have a stronger threshold stimulus, you could start another action potential. But it will fire, it'll have a smaller amplitude, which means it won't be as high. Some of the sodium ion channels have closed in order for this to occur, and there are still a few potassium open. This also helps to ensure one-way travel, because that's really important. If you had messages going both directions, they would cancel each other out and it wouldn't work. All right, so impulse conduction. We talked a bit, and I think I might have even mentioned saltatory conduction. An unmyelinated, no myelination axon conducts impulses all across the surface. Okay, so it's got to go all the way across the axon. Myelin, remember, is a fat, and it's rich in fat and its lipids, and it acts as an insulator, and it prevents the flow of ions through the membrane that it is covering. Okay, and so we have these gaps that I pointed out when we looked at the diagram of the neuron. And we have these gaps here, and we call those nodes of Ranvier. They're in the sheath. They interrupt the myelin, where the channels can open and close still there. All right, so you can have a little bit of channels open and closing in the middle. In a myelinated axon, the current basically is going to jump from one to the next. It skips over that little section. I didn't do it quite right there. Should have gone here. Well, I did that one right. Never mind. So yeah, it's going to skip from one to the next. All right. So this is what we call saltatory conduction. They're transmitting impulses, and it is faster because it can skip over those sections. So if you take a shortcut, it's faster. If you can skip certain parts, it's going to be faster. And so saltatory conduction, myelinated axons transmit impulses faster than what we call a thin axon fiber, which would mean without any myelination. There are many diseases like multiple sclerosis, which affect the myelin sheath and therefore nerve conduction. And so I told you earlier, and I should go get the cord for my other cell phone cord, an old one, but you know how they get frayed, and we're still using them because, you know, we all get irritated with these cell phone cords when they don't charge. But when they get frayed right here, what happens? It starts having an interruption in charging, right? It'll work and not work. So think about that occurring to things like your muscles. All right, so if you're trying to send a message to your muscle and it keeps getting interrupted and you're trying to move forward, you know, you might have a stutter there, okay? And so diseases like multiple sclerosis and many others are some of those that are caused by loss of what we call the myelin sheath. This is a really good slide that summarizes everything we just talked about. So be sure that you study this and that you know these steps and which order they occur. I really highly recommend drawing them out, labeling the ions and what happens. So let's look at what happens. Let's apply this clinically. What can happen when there are problems here that can affect impulse conduction? You can have paralysis, convulsions, and other things result from changes in the permeability of ions in axons. Okay, so how well they're allowed in or out. Several substances can affect ion permeability. Things can increase your potassium permeability, and that can partially cause a depolarization in the membrane. This can cause the threshold potential to be reached with a smaller stimulus. So it can cause an action potential to occur more easily. The affected fibers are often excitable, and you can have convulsions occur, so it can cause a convulsion. Decreases in potassium, the resting potential becomes more negative, and the action potentials then it's harder for the action potential to be fired because that distance between them is greater. And so muscles can actually become paralyzed. It can cause paralysis. Okay, paralyzed, that's not a word. Paralyzed. Anesthetic drugs, all right, work using this type of information, I should say. These decrease membrane permeability to sodium, which prevent impulses from passing through to the brain, and that stops the perception of touch and pain. So understanding these is not only important for treating different occurrences, but also for ways that we use different medications. And we'll talk about that a little more when we look at the synaptic cleft and the serotonin reuptake inhibitors.